Hello and welcome to the GCSE Biology tutorial. This covers topic B2 organisation, and this tutorial is part 1, animal gaseous exchange and animal digestion. So we'll be looking at how cells are organised, lungs and gaseous exchange, enzymes, digestion and food tests, and 2 practicals including enzyme activity and food tests. Starting off with, you need to know that cells, which we've talked about before, are the building blocks of life. And they become differentiated and can form different cells. So there's not just one kind of cell. And groups of cells working together are called tissues, for example, muscular tissues. And then tissues work together inside organs, and then organs work together in an organ system. And this makes up an organism. There's an example there of the circulatory system, which is made of the organ, the heart, which is made of tissue like heart tissue, which is made of heart muscle cells. So starting off with the lungs are in the thorax, which is the upper part of the body, and underneath the lungs there's a muscle called the diaphragm, and the rib cage and the pleural membrane surround and protect the lungs. When you breathe, air comes into your nasal cavity and down the trachea, and then into the bronchi, which is 2 of, and then branches again into the bronchioles, and then finally into those tiny air sacs called alveoli. We've looked at those before. But here's a close-up of the alveoli, and this is where gas exchange occurs. So oxygen moves into the blood from the alveoli, and carbon dioxide passes out the blood into the alveoli and then into the air. So what are enzymes? Enzymes are biological catalysts. That means they speed up a reaction and don't get changed themselves. They're actually proteins, so they're made of amino acids, all folded into a unique shape. The way in which they work is using the lock and key model, which I'm going to go through here. So, an enzyme has a particular area called an active site, and that's the area that the substrate or the molecule you're trying to break down connects with. It's a bit like a lock fitting in with a key. If it fits in correctly, the enzyme will then break down the substrate and be free to carry on and work again. Different enzymes have different roles, so different enzymes have different shaped active sites, and that's why it's called the lock and key method. Remember, we call the molecule that fits into the enzyme the substrate, and we call the area that it fits into the active site. What conditions do enzymes prefer? Well, temperature. First of all, if it's a hotter temperature, enzymes work better. But if it gets too hot, that active site changes shape, and the enzyme becomes denatured because the substrate can't fit in there anymore. And this is temperatures after 40 degrees. So the best temperature for an enzyme to work is below 40 degrees, not over 40. In terms of pH, the best pH depends on what the enzyme is. So for example, enzymes in the stomach work best at pHs of 1 or 2. Enzymes in the mouth work best at pHs 7. But again, the best pH is called the optimum pH. You'll need to know about a practical where you investigate enzyme activity. In this practical, you're looking at how pH affects enzyme activity. The enzyme we're looking at is amylase, and that enzyme breaks down starch into a simple sugar. You can test that with iodine. So iodine is orange and then changes to blue-black if it's in the presence of starch. The first thing you would do is you'd add to a spotting tile drops of iodine, as shown in the diagram. You then need to create your solution of amylase and a buffer at a certain pH, so for example buffer pH 5. You then add starch to the amylase. Once you've done this, every 30 seconds you would add drops of this amylase and buffer and starch solution to the iodine. Now what will happen, it will turn blue-black because it's showing that starch is present. But when the enzyme starts to work and the amylase gets broken down, the amylase breaks down the starch, there won't be any starch left. So when the iodine remains orange and doesn't turn blue or black anymore, it means the enzyme amylase has broken down the starch. So you would time how long this would take. And then you can repeat it with different pH buffers, making sure you keep certain things the same, like temperature by using a water bath, and the concentrations and amounts of the solutions. Let's look at those enzymes in a bit more detail. Carbohydrases. These change carbohydrates into simple sugars. An example of a carbohydrase enzyme is amylase. Proteases. These change protein into amino acids. Lipases. They change lipids and break them down into fatty acids and glycerol. Bile is also important in digestion, but it's not an enzyme. Bile is made in the liver and stored in the gallbladder, and it neutralises the stomach acid when it goes into the small intestine, and it also emulsifies or breaks down fats. If you pause the tutorial now and answer these questions. So, how are organisms arranged? Cells into tissues, into organs, into an organ system. How do enzymes work? You need to talk about the lock and key model and mention the fact that enzymes have this unique active site which the substrate fits into. Denatured. Now, it doesn't mean the enzymes died. It means the active site has changed shape, so the substrate no longer fits. So the enzyme doesn't work. What does the enzyme lipase break down? It breaks down lipids, or fats, into fatty acids and glycerol. Moving on to what actually happens in the digestive system. In the mouth, you have salivary glands, and they produce a liquid called saliva, which breaks down the food. But it also not only softens it, it contains an enzyme, and that enzyme is amylase. So that actually breaks down starch in your mouth. The food then passes down the esophagus, and this contracts with muscular contractions to move the food along. The food then lands in the stomach. The stomach has enzymes in it, such as pepsin, which is actually a protease, which breaks down protein, and the enzyme contains hydrochloric acid, which kills bacteria and gives the right pH for the enzymes. The food then moves into the small intestine, and that's where more enzymes break down the food. And the digested food is then absorbed through the small intestine, through the villi, into the blood. So that's the most important part of digestion, the small intestine. Anything that doesn't get broken down moves into the large intestine, where all the excess water is absorbed back into the body. This waste is then stored in the rectum before being passed out of the anus. There's two other organs important here. The pancreas, that produces enzymes, and the liver, which produces bile. Food does not pass through these organs, but they are part of the digestive system. You also need to know about food tests. So how would you test for certain foods? Now, to do this, you would need to prepare your sample by grinding it down in pestle and mortar and mixing it with water and filtering it so there's no solid bits inside. The test for glucose involves adding Benedict's solution. You would add the Benedict solution to your sample and then heat in a water bath and leave. And if glucose is present, you will see a colour change of blue to green to yellow to brick red. To test for starch, you would add iodine. Iodine is orangey brown and it will turn blue-black if there is starch. To test for protein, you add burette solution and shake gently. If there is protein, it will change to a purple colour. To test for lipids, you would add Sudan 3 stain solution and again shake gently. If there is lipids present, the top layer will go bright red. You can also test for lipids by placing the food substance on filter paper and looking for a transparent residue or an oily residue. If you pause the tutorial now and try this 6-mark question to describe how enzymes are used in the digestive system. Give yourself 10 minutes. So, how are enzymes used? Well, first of all, digestive enzymes, the whole point of them is to break down foods, nutrients and molecules so they can be absorbed into the small intestine and the blood. So what does that happen? It happens, first of all, in the salivary glands in the mouth. Amylase, that breaks down starch. You've then got the small intestine and the pancreas. They produce enzymes such as protease, which break down protein into amino acids, carbohydrates, which break down starch into simple sugars, an example of which is amylase, lipase, which breaks down fats into fatty acids, and glycerol. The stomach produces the protease enzyme called pepsin, which breaks down protein. And you can also mention bile. Bile is not an enzyme, but it's made by the liver to break down lipids by a process called emulsification. We've covered cell organisation, lungs, enzymes, digestion, food tests, and enzyme activity. Thank you.